Hi guys, in this PowerPoint slides, I will explain one case study of positive and negative regulation of transcription, page 522 in the book. I will discuss one example of yeast cell, and I will show you how yeast cell will use a specific type of monosaccharide known by galactose as a source of carbon instead of glucose. This example is very similar to the lac operant discussed in the handout. إذا بتتذكروا كنا نقول in the presence of glucose E. coli, the bacteria E. coli, you will use the glucose as a preferred source of carbon. However, in the absence of glucose and in the presence of lactose, the cell, the E. coli or the bacterial cell, will start to express specific type of enzymes ready to metabolize the lactose. So first we will convert lactose into glucose, and then you will use the glucose as a source of carbon. نفس الكونسبت رح يكون applied here in eukaryotic cell in yeast. رح ناخد هالمرة example of galactose sugar. مثل ما حكينا عن البكتيريا yeast cells prefer the glucose as a source of carbon. But in case the glucose was absent, شو بتعمل yeast cell? The yeast cell will search for another source of carbon and another source of energy. هالمرة will search for another sugar known by galactose. In order to metabolize the galactose, the yeast cell will start to express enzyme, to produce enzymes in order to metabolize the galactose as a sugar. So inside the genome of a yeast cell, you will find a specific set of genes. It's not an operon لأنه اتفقنا بالintroduction no operon in eukaryotic cell, but you will find a set of genes in the genome of the yeast cell ready to be expressed into enzymes only when the galactose is present and the glucose is absent. So guys, بدي منكم تقطعوا معي على هيدا السلايد. I will start first by the yeast cell ورح نقطع ب two pathways. In the first one, if glucose is present, the galactose gene or the gal genes are off. So it's a regulated gene. No need to transcribe the genes encoding enzymes that metabolize the galactose because the glucose is the preferred source, so the gal genes will be off. However, if the glucose is absent, galactose also is absent, okay? Also the gal genes will be off. However, if glucose was absent and the galactose is present in the medium, now you will turn on the galactose genes or gal genes. You will start to produce the enzymes required for the metabolism of galactose. So هيك لناخد بس brief guys عن the process. إذا بتلاحظوا here we have in this slide a yeast cell. Galactose first is present outside the cell. It's an extracellular galactose. First, we need to transport the galactose from outside to the inside of the cell. So we need a protein known by Gal2 protein produced by a gene known by Gal2 gene. This protein acts like a transporter protein located at the level of the membrane of yeast cell. So here at this level, okay, at the level of the membrane. So the galactose will be transported from outside to inside. Once inside, the galactose intracellular in contact with an enzyme known by GAL1 enzyme produced by GAL1 gene. It's a galactokinase. Transform this galactose into galactose 1-phosphate. It's a kinase, so add 1-phosphate group. Then the galactose 1-phosphate with another enzyme known by GAL7, it's a galactose transferase, transform the galactose 1-phosphate into UDP galactose. It's a nucleotide sugar. In addition, UDP galactose, in the presence of an enzyme known by GAL10, it's a galactose epimerase. It will be converted into UDP glucose. Don't forget that galactose and the glucose are isomers. Then UDP glucose plus GAL7 enzyme, it will be reconverted into glucose 1 phosphate, and the glucose 1 phosphate now is present inside the cell and ready to start the process of glycolysis, finally leading to the production of energy or ATP. إذا لاحظتوا guys, all these genes 1, 2, 7, and 10 should be only transcribed when galactose is present. If galactose is not present, this signaling pathway, the signaling cascade will be turned off. All the genes will be turned off, and the cell will use the glucose instead of the galactose as a preferred source of carbon and energy. This is a Between enzymes guys, the function and the signaling pathway انتم مش مسؤولين عنها, so you have only you need only to understand what is the function of each of this enzyme, but without memorizing it, okay? In this slide, you can find the presence of chromosome 2. It's part of the yeast genome, where you can localize the GAL1 gene, the GAL10, and the GAL7. 1, 10, and 7 genes are located near each other but do not constitute an operon. Each gene has its own promoter and its own termination sequence. On another chromosome, chromosome 12, you can find the GAL2 genes responsible in the production of the GAL2 protein transporting the galactose from outside the cell to the inside. بس انتو guys مش مسؤولين تدرسوا الجال 2 مثل ما موجود بالبوك وباقي السلايدز. نحن will focus only on GAL 1, 10 and 7. Let's start by one example, the absence of glucose. مجرد ما يختفي الجلوكوز من الميديum surrounding the yeast cell, a gene known by GAL4 gene will produce a protein, regulatory protein, known by GAL4 protein. The GAL4 protein is produced only in the absence of glucose and act as an activator. So the GAL4 protein, the function of this protein is an activator. درسنا قبل إنه the activators will bind to enhancers in order to stimulate 100-fold the process of transcription initiation. So in this case, the GAL4 will bind to a sequence known by UAS, upstream activating sequence, representing enhancers. One UAS is present for both GAL10 and GAL1, and one UAS for the GAL7. So the GAL4 protein, the activator, the regulatory protein, will bind to its DNA binding site known by UASG for upstream activating sequence for galactose, representing an enhancer. خلينا نحكي more about the GAL4. GAL4 is a regulatory protein activator. إذا بتتذكروا بالفيديوز اللي بعثت لكم اليوم من قبل حكينا more about the activators وقلنا activators are present either as monomer or as dimer, homo or heterodimer. In this case, the GAL4 activator is present as a dimer. It's a homodimer with 3 domains. نحن تذكروا قلنا each activator has at least 2 key domains. The DNA binding domain, so هيدا هو أول واحد, allowing the GAL4 to bind to the UASG. So this is the DNA binding domain, okay? And we have an activation domain allowing the recruitment of the co-activators, and we will continue that process مثل ما شفناه هون قبل. And in this case also we have a third domain known by dimerization domain. Why it's the dimerization domain? It's present here in the middle because it's a dimer, so we need to join the first monomer with the second one to form a complete dimer. So بلشنا guys إذا بتتذكروا in the first case the absence of glucose. In the absence of glucose, the GAL4 is produced and bind to the UAS. But if the galactose is absent, the activators should be switched off. اوكي سو انا اذا الجلاكتوز مش موجود انا هالاكتيفيتور ما لازم شغله سو لازم اطفيه. How I can turn off the GAL4 activator? By the production of a repressor known by GAL80. So in the absence of glucose and in the absence of galactose, GAL4 is produced, but at the same time, a gene known by GAL80 will produce a repressor protein known by GAL80 protein. The GAL80 protein will bind to the GAL4 and repress it. So, as the final result, you will find an inactive GAL4 due to the presence of the GAL80 repressor with no galactose. However, let's consider now that the galactose is present in the medium. So the glucose is absent, and now you will find the presence of galactose in the medium surrounding the yeast. In the presence of the galactose acts as an effector. يعني بمجرد ما ينوجد رح نبلش نشغل الجينز اللي هن GAL1, 10 and 7 to metabolize the galactose. So in the absence of glucose and the presence of galactose, a gene known by GAL3 genes will produce a protein known by GAL3 protein. This protein will bind to galactose. Okay, so the GAL3 protein produced will bind to galactose and converts the galactose into an inducer. This inducer will bind to the GAL80. شايفين ال GAL80? So the inducer will bind to the GAL80 and dislocate means change the location of the GAL80. So the GAL80 is no more attached to the activation site of the GAL4, and the GAL4 will be converted from inactive to active. Now the activator is active, so can start the transcription initiation of GAL1, GAL10 and GAL7. What does the GAL4 activation domain do? رح نرجع لفيجر already شفناها من قبل بالبوك. إذا بتتذكروا this was the structure of the gene. Here you will find the different GAL genes, the theta box at the level of the core promoter. Facing the core promoter you will find the enhancer here, it's known by UAS. So upstream activating sequence for galactose, where you can find the binding of the GAL4. It's an activator, so recruits in co-activator. تتذكروا سمينا the co-activator in the case of yeast cell a mediator. هي هون بنلاقي المediator. The mediator will recruit an RNA polymerase 2, and at the same time at the level of the theta box, the general transcription factors will start to assemble, starting by the transcription factor 2D, then A and B, the RNA polymerase with F, with the mediator and the activators will join here at the level of the core promoter, the general transcription factors, and we will finish by the E and H. Here the complete general machinery is produced and you can start the transcription initiation of the GAL genes. Point number 3, انتو مش مسؤولين عن هالترم guys اللي هو السوي SNF، بس بدي منكم تعرفوا شغلة انه the GAL4 activator رح نشوفها later بغير lectures will recruit some enzymes responsible in the remodeling of the nucleosome or the chromatin structure, converting the chromatin from 30 nanometer chromatin fiber to 10 nanometer chromatin fiber. معناتها كأنه فتحنا الchromatin, so the chromatin at the level of the data box or the promoter now is more accessible by the general transcription machinery, and you can start the process of transcription of the GAL gene. In addition guys, خلينا نبلش بالكيس. نحن اللي قبل شوي شفناه هون هو in the absence of glucose than in the presence of galactose. خلينا نرجع للأساس. Let's consider we have a case in the presence of glucose. كلنا بنعرف وقت الجلوكوز يكون موجود, it's the preferred source, main source of carbon used by the yeast. معناتها will turn off the expression of any other genes required for the metabolism of any other source of monosaccharide or sugars. معناتها هون we will focus only on glucose. So when glucose is present, the cell would rather use that so the galactose genes are repressed even if the galactose is present. So if and at the same time we have glucose and galactose, the cell will select only the glucose and turn off or repress the galactose gene. So you have to keep in mind that glucose in the yeast GAL gene system exerts a catabolite repression. So مجرد ما يكون موجود الجلوكوز كل شيء تاني رح ينطفى أو it will be switched off. How the glucose will exert a catabolite repression? In this case, a gene known by MIG1 gene will produce a protein known by MIG1 protein. This protein will bind to a DNA site known by MIG1 site or URSG for upstream repressing sequence for galactose. تشبه ال UASG بس ال UASG was the upstream activating sequence بينما ال MIG1 site is the URSG for upstream repressing sequence for galactose. MIG1 protein is a repressor, binds to the URS, and using its repression domain, the MIG1 will cover the activation domain of the GAL4 at the level of the UAS and block its action. So in the presence of MIG1 protein, the repressor, the GAL4 is not active anymore and you will switch off the expression of the GAL1 genes into protein. وهيك the metabolism of the galactose also will be switched off and the cell will use only the glucose as a source of carbon and energy. In addition to MIG1, the repressor, sometimes you can find the TUP1. TUP1 is a co-repressor recruited by the MIG1. This TUP1 will function as an HDAC. It's an histone deacetylase بنشوفها بعدين. هيدي الانزيم guys رح تساعدنا to convert the DNA, the chromatin from 10 nanometer to 30 nanometer. كلنا بنعرف when the chromatin is present in its 30 nanometer, the structure will be compact, condensed, so the transcription is off. بهيدي السلايد عم فرجيكم the difference between a 30 nanometer chromatin fiber where the genes are inactive and the open configuration, the active genes are present in the 10 nanometer chromatin fiber. So to end these slides guys, in sum, what we can conclude from this case study. In sum, GAL4 acts as a positive regulator, so it's an activator. GAL80 protein acts as a negative regulator, so it's a repressor. While the galactose, it's an effector molecule. Why it's an effector? Because in the presence of galactose, this molecule will bring about a certain effect, will allow the GAL3 to convert the galactose into inducer. The inducer will dislocate the GAL80 from the activation domain of the GAL4, activating the activators, and the GAL genes will start to be expressed. Okay, so in general, this is the example of galactose utilization in yeast cell. إذا لاحظتوا, it's similar to the case of lac operon. These genes are considered as regulated genes because they are expressed only when needed, بمعنى تاني in the absence of glucose and in the presence of galactose.